Agrobacterium tumefaciens infects the plant through its T plasmid. The T plasmid integrates a segment of its DNA, known as tDNA, into the chromosomal DNA of its host plant cells. A tumefaciens has flagella that allow it to swim through the soil towards photoassimilates that accumulate in the rhizosphere around roots. Some strains may chemotactically move towards chemical exudates from plants, such as acetylcyringone and sugars, which indicate the presence of a wound in the plant through which the bacteria may enter. Phenolic compounds are recognized by the Vera protein, a transmembrane protein encoded in the Vera gene on the T plasmid. Sugars are recognized by the Ch protein, a chromosomal gene-encoded protein located in the periplasmic space. At least 25 Vera genes on the T plasmid are necessary for tumor induction. In addition to their perception role, Vera and Ch induce other Vera genes. The Vera protein has autokinase activity. It phosphorylates itself on a histidine residue. Then the Vera protein phosphorylates the Verg protein on its aspartate residue. The Verg protein is a cytoplasmic protein produced from the Verg T plasmid gene. It is a transcription factor, inducing the transcription of the Vera operons. The Ch protein regulates the second mechanism of the Vera gene's activation. It increases Vera protein sensitivity to phenolic compounds. Attachment is a two-step process. Following an initial weak and reversible attachment, the bacteria synthesize cellulose fibrils that anchor them to the wounded plant cell to which they were attracted. 4 main genes are involved in this process: CHV, CHVB, PSCA, and IT. The products of the first 3 genes apparently are involved in the actual synthesis of the cellulose fibrils. These fibrils also anchor the bacteria to each other, helping to form a microcolony. Verg, the most important virulent protein, is a necessary step in the recombination of illegitimate recolonization. It selects the section of the DNA in the host plant that will be replaced and it cuts into this strand of DNA. After production of cellulose fibrils, a calcium-dependent outer membrane protein called ricadhesin is produced, which also aids in sticking the bacteria to the cell wall. Homologs of this protein can be found in other rhizobia. Currently, there are several reports on standardization of protocol for the agrobacterium-mediated transformation. Effect of different parameters like infection time, acetylcyringone, DTT, cysteine have been studied in soybean.